Very excited about the opening plenary session, which is actually going to be the first of two sessions exploring multiple potential roads to conscious experience. Today we're going to be hearing about global neuronal workspace theory, predictive coding, and quantum approaches to consciousness, and we're fortunate to have three preeminent speakers in these fields. The first is Dr. Dehaene, who is a professor at the Collège de France, where he is also the director of the Cognitive Neuroimaging Laboratory. Professor Dehaene is internationally renowned for his work in numerical cognition, the neurobiology of language, and of course, consciousness. Please join me in welcoming him.
Well, thank you very much. It's a great honor to be here today and to give this lecture. My path to consciousness started about 18 years ago when Jean-Pierre Changeux and I, also with Lionel Nakache, following up on leads by Bernie Barth, proposed a specific hypothesis called the global neuronal workspace hypothesis. And I'd like to review with you today the status of this hypothesis, show you a lot of empirical data that seems to go in the same direction. The idea was very simple. We know that the brain is comprised of many specialized processes, such as face recognition, sound processing, respiration monitoring, so on and so forth. And our idea is very simple. There is a system that allows for the breaking of this modularity. What we subjectively experience as consciousness is the global availability of information, which allows it to be shared, both with us and with others. So there is a system that takes information from one of the modules, as shown on these slides. There is, of course, a series of visual modules, for instance, that carry visual information upwards in the hierarchy. But at some level in the hierarchy, we reach a system which is able to broadcast the information widely, share it with other modules so that we can flexibly reorient, route the processing to many other processes. And so our idea is that consciousness relates to the activity of a global neuronal workspace, which has evolved specifically for this function of broadcasting a specific piece of information which has been selected. I could show you a lot of data on the anatomy of the system, but let's just think of it as a set of high-level areas, not a localized system, but specifically involving prefrontal cortex, parietal cortex, high-level associative cortex of the temporal lobes, working together, linked through long-distance connections, able to share the information at the scale of the brain. And there is a specific dynamic property of this system because it is recurrently connected. It is showing an ignition threshold. So we think that conscious access corresponds to the ignition of workspace neurons coding for the selected content. This is just a very specific cartoon view to show you what might happen when you're flashed with a piece of information, depending as whether it is above or below the threshold of consciousness. You may see the information progressing through a series of processes, but if it is above the threshold for consciousness, it's going to reach to high-level areas of the brain, including prefrontal cortex, start a reverberating assembly in this high level, a stable or so-called metastable assembly, which is able to sustain activity and send activation back to the areas that started it. On the other hand, if the stimulus is below threshold, as illustrated on the left part of the slide, it's going to advance through the processing stages, but it's not going able to reach this ignition threshold where it's going to create a global assembly. So one property of consciousness is that you're able to sustain the activation for as long as needed to sustain a particular goal, regardless of the actual duration of the initial stimulus. So I want to illustrate in this talk three predictions of the global neuronal workspace and how they are held in data. First, I'll appeal to the masking stimulus, a situation where you flash a digit, for instance, in my case, follow it by a mask made of letters, and depending on the length of the presentation, you may or may not be able to become conscious. So this is one way to manipulate consciousness experimentally. And the prediction is very simple. Initial brain activity should be unconscious. Conscious perception should involve the late global brain scale synchronous waves of activity engaging high-level cortices. Then I'll show you some data from dual tasking, where we ask people to do two things at once. And because this workspace is acting as a central limiting resource, the prediction, very simply, is that if you do one task consciously, then this will be blocking conscious access for another task. We should be able to see that performance of a first task impairs consciousness of a second. And again, the initial processes should operate unconsciously for either task, but at the central stage, only the late global brain scale activity should be delayed for the second task. Finally, I will speak about spontaneous thought. I don't think it's necessary to introduce external stimuli in order to study consciousness. According to the workspace model, there is spontaneous activity in this workspace system, so that even in the absence of external stimuli, it's running on its own, it's generating its own patterns of activity. And I'll show you at the end ways in which we can extract such evidence from resting state data, and show you that resting state, when it is conscious, corresponds to a dynamic series of global brain scale states, whereas under unconscious situations such as anesthesia, this is no longer the case. So this is where I do my work, the Neurospin Center in France, and we have, of course, a number of tools at our disposal: functional MRI, event-related potentials, even in young babies, magnetoencephalography, and we analyze intracranial recordings. I'm showing this just to show that I will insist on the later techniques of electroencephalography, magnetoencephalography, intracranial recordings, because they give us access to the dynamics. And according to what I'm telling you today, it's all in the dynamics. You can separate non-conscious and conscious stages of processing through the dynamics. In particular, I want to make use of a technique which I think has a lot of potential, which is called multivariate decoding of brain signals, and we apply it to MEG and EEG signals. It's a way to essentially take the dynamics of the signals and project back into the psychological domain, asking what can we decode from brain signals. So the way we apply it is we take, for instance, the 306 sensors of magnetoencephalography that we have, plus the EEG channels, and we take a time slice of the data, so a lot of sensors, and we ask, can we build a decoder of this brain activity, which tells us what the subject had in mind, what was the stimulus, whether the subject was conscious or not. We do that by sliding this window of time. We get a time curve like this, which tells us that before the stimulus, there is no way to decode what's happening, but after the stimulus, after time zero, for a short while we may not be able to decode, and then suddenly we find information in the brain. And I show you that we can derive from brain activity a lot of different pieces of information, some of which is unconscious and some of which is conscious. So we get the dynamics of activity. We essentially can tell for how long a given representation is present in the brain using this technique. But because we are training different decoders at different time, we get information about different brain areas which are responsive to a particular parameter, and we can ask how this is generalizable across time. And I will show you how useful this technique is to ask how long a representation is lasting in the brain. If it's a chain of processes, as we think is occurring unconsciously, then we should see a lot of diagonal processing. That is to say, we can train a decoder, but it does not generalize far in time. But if there is a metastable assembly in the brain, as we think is occurring during conscious processing, then we should be able to see it as a sort of square pattern of generalization. We train the decoder at one time, we can generalize to another time point. So we think that this is a highly diagnostic technique that tells us what is the representation, how long it is lasting in the subject's brain, whether it is stable or not. So this is very abstract. Let me immediately give you some examples. This is the masking paradigm we've been using. I was hoping to show it to you, but I was not able to use my own computer, so I'll just tell you how it works. Flash a digit, we have a lag, which is variable, and then we flash some letters around the digit. If the paradigm is right, if the delay is short, you don't see the digit at all. You just swear there was no digit. So it's completely unconscious. And then as you make the duration, the delay between the digit and the letters longer and longer, suddenly you begin to see the digit. And there's a very sharp threshold for subjective experience. So suddenly, really we can speak of a threshold of consciousness in this paradigm. You begin to see the stimulus. We've been doing many experiments with this paradigm. I'll show you a few, but this is the basic result, that we can distinguish between linear unconscious processing stages versus nonlinear conscious stages of processing. So if we look at the profile of activation in different brain areas, initially it is essentially linear with the stimulus. I hope you can see that. I don't know if I can point on this screen. Maybe I can. Can you see a pointer? No. Well, I hope you can see here that in the visual regions of the brain, you get this very clear pattern of activation which follows two stages. And the first stage is activation essentially climbing linearly with the stimulus. And this is completely unlike what the subjects report. In particular, you get quite high level of activation in occipital and temporal areas of the brain, even on trials where subjects report not seeing the stimulus. So this is unconscious activity. Now, if we look for traces of conscious activity in the brain, we find it much later in processing. We find it in the second wave. You can see that in the second wave, all of the short durations of presentations have collapsed to zero. And this coincides with the subject's reports of not seeing the stimulus. When the subjects report seeing, we see this much more intense and amplified wave of activation that remains for a long time. And I remind you that the stimulus is gone for a long time. The stimulus is only 16 milliseconds long, but the brain is able to hang on to that stimulus. It seems to occur primarily in high-level areas such as prefrontal cortex, but at the same time we see a return of the activation in the original occipitotemporal areas. This two-wave pattern is quite characteristic, two-stage, first stage linear, second stage nonlinear. Here is how it looks like with our decoder. So we train a decoder to tell whether the stimulus is present or is not present, the digit is present or not present. We train the decoder at the longest duration of 100 milliseconds, and then we see how well it generalizes to the earlier periods. And what you can see here is that even in the earlier periods, like 33 milliseconds of delay between the digit and the letters, a duration where you don't see the digit, we can decode it. We can decode it from the brains of people who have not seen it. They swear there was no digit, but we can see it's there. But what you can see is that the pattern of activation is quite different from when people report seeing it. If we look at the different time points of these decoders, each of these curves here corresponds to one particular decoder, we can see that the early decoders that are getting data from early on in brain activation, they essentially decode all of the delays between the digit and the letter. If we move slightly later on, we can see a linear pattern of activation. It's only quite late in the epoch that we see that activation returns essentially to zero, decays to zero, in the cases where subjects are not conscious of the stimulus, but stays very stable with this sort of square pattern which indicates stability when the subject is conscious of seeing the stimulus. The square pattern, or sort of thick diagonal, indicates that although brain activity is still evolving, it's also evolving at a much slower pace. It's not this sharp diagonal, it is essentially a metastable pattern of activity lasting for a long time in the brain. And we get this sort of evidence even if we pick the very same physical stimulus. I think this is the best paradigm we can have. We have the same physical stimulus, but subjects report seeing or not seeing on different trials. You can see that what distinguishes them is this late form of activity, the square pattern. It starts around 200 milliseconds, begins to be much more amplified for the stable conscious representation, and then you see this square pattern of activity. So what we are seeing is that metastability is a correlate of consciousness, and there's a lot of work by Aaron Scourger in other paradigms suggesting that this is the case, that if you have a stable pattern of activation, it seems to relate to a conscious perception. This decomposition of early linear versus late nonlinear activity extends even to new conditions such as the baby's brain. A few years ago, with Sid Quider and my wife Ghislaine, we asked, could we generalize these paradigms to the baby's brain? And of course, we cannot flash digits. They are not very interested in digits, but we can use faces. We flash a face, sandwich it between masks such that at short durations you don't see the face. And then we record from brainwaves of the babies, and what we saw just in one slide is that the same sort of decomposition of two stages or two sorts of stages is found in the baby's brain. Early on you find linear waveforms that really show the linear duration of the stimulus. But if you look at the late stages, all of the short durations of presentations return to zero, and all of the long durations of presentation deviate from zero and indicate a stable pattern of activation. It turns out that the threshold in question, which is around 100 milliseconds, is also the threshold for the baby to orient towards the face. And so it coincided with a behavioral threshold being able to use the visual information about the face. So it really looks as if the baby is organized just like the adult, and this is true of even 5-month-old babies. I'm showing you the 12-month-old data here. What it seems to suggest is that babies are already having this decomposition of non-conscious and conscious stages of information processing. Only I draw your attention to the slowness of this phenomenon. The nonlinear ignition seems to be occurring 3 times slower at least in babies compared to adults. So they access consciousness, we think. They have all the signatures of consciousness, but they do so much slower. Well, one direction has been to extend this work to babies. Another direction has been to try to get more specific about the areas that are responsible for this ignition. And one way is to go intracranial, use the ability we have to record inside the brains of epilepsy patients from electrodes. And using this and presenting unconscious information, we've been able to replicate this notion of an ignition late in only in the case of conscious stimuli. So here you see what happens to a non-conscious word and to a conscious word when we flash it and we track its activation through many intracranial electrodes across about 10 patients. You can see that the activation starts in the occipital cortex, and there it's about the same for conscious and non-conscious stimulation. Progresses forward into the occipital temporal cortex, continues to progress, and it's only by around 300, 400 milliseconds that you see this expanded pattern of activation expanding into the frontal lobe. You see this extremely clear difference in high gamma and also event-related potential activity in the frontal cortex only when the subject reports being conscious of the word. If the subject reports not being conscious, you get the early activation but not the late one. Another signature of consciousness in this paradigm is found in the time-frequency domain, where we analyze the frequency of the responses and not just the time. So time is on the x-axis, frequency is on the y-axis here. And what you can see is several additional signatures of consciousness. First, there is the prolongation of high gamma activity, which you can see here on the bottom, and the presence of this lower alpha and low beta suppression of power, accompanied by beta synchrony across many areas. What this means is that activation gets prolonged. You can find some gamma activation unconsciously, but it gets strongly amplified, and it gets longer lasting, and the brain starts to communicate with itself with these phase synchrony measures, as well as also Granger causality measures. What you see is an enormous amount of exchanges of information occurring late in the epoch after 300 milliseconds, being a signature of conscious perception of a non-conscious, of a flashed, masked word when it crosses the threshold of consciousness. There are many paradigms that have cross-validated these notions. I want to show data which is not from my lab, but from the beautiful work of Rodrigo Cue and Quiroga. Rodrigo flashed some pictures. He was able to record from single neurons in humans, using also the expertise of Itzhak Fried. And what they showed was that you can find single neurons that are both content-specific and consciousness-specific, in the sense that you see this neuron, which likes the World Trade Center, fires only on unique trials where the subject is reporting C in the picture. The picture is flashed, the picture is masked, so that on many trials, subject reports not being able to see the picture. You see that the spikes of the neuron, which are here, the color, which is blue, indicates that these are trials where the subject was able to report the picture. If the subject is not able to report the picture, these are the red trials, you see no spikes. And on average, across many neurons, you may see a little bit of a transient spiking for an unconscious stimulus, but you see a sustained firing, much larger initially and then sustained for a conscious stimulus. It's possible to pursue this even further down into non-human animals, and I want to mention the work of Victor Lamme and Peter Rufzimmer. We have been showing that indeed, again, conscious perception relates to a late wave of amplified activity. This is in V1, this is multi-unit spiking activity, and in their stimulus is a shape that can be sort of popping out from the background. Only when the animal reports seeing the stimulus popping out from the background do you see this late wave of amplified activation. You don't see it in case of not seeing, and although you can see that the initial activation is essentially identical. So strong initial activation, but seeing correlates with late amplified activity. Neuroscience is also bringing this down to actual mechanism, and here I want to mention the beautiful work of Matthew Larkham, which is suggesting one way in which ignition may work. There is a mechanism called backpropagation activated calcium spike firing, backfiring, which is extremely interesting. It's a situation where you get amplified spiking, as you see here in the bottom. Looks very much like our nonlinear ignition. You get it specifically when you get a conjunction of inputs to the same pyramidal cell impinging on the apical dendrites of the cell of these layer 5 neurons. And at the same time as you get inputs closer to the soma. Then you get an enormous amplification of the firing. And remarkably, Matthew Larkham, together with the Riken group of Manita Murayama, have been able to show how this plays out in an actual perception task. They were not using vision like we were using, but they were using touch. And they designed a situation where you can record a touch stimuli in the mouse. This is very interesting that this is now getting to actual mechanism in the brain of the mouse. In the mouse, when you present a touch stimulus, you again get this biphasic response of somatosensory cortex. And in between, you get this activation of a much more anterior premotor area, M2. What they found is that there are direct top-down connections from M2 to somatosensory cortex. They wonder whether these could be responsible for this second peak of activity, and indeed they are. When you inactivate the area M2, you lose specifically the second peak, and you keep the first peak, which is bottom-up, and you lose specifically this mechanism of backfiring has been identified by Larkham. So disruption of the M2 activity blocks the late large calcium spikes that you get on the apical dendrites of these layer 5 neurons in area S1. So it is this actual mechanism which seems to be responsible for this late firing. And the last bit of proof in this paper, which is so interesting, is that the mouse in which we block these top-down inputs, the mouse is no longer able to use touch information actively in order to navigate and avoid one room and choose another room based on touch alone. So this is a case where you have proof by interfering with brain activity that interfering with the late wave of activation interferes with actual perception, usable perception in the mouse. I think we're going to see many more such studies in the future, but they all point to this notion of biphasic activity with the late phase correlating with reportable, usable information. Now, we may also use these paradigms to ask difficult questions, such as the function of being conscious. There are some people who have been saying that because it's so late, 300 milliseconds, almost half a second, maybe there is no particular function to consciousness. It's too late. It's after the fact. But I don't think so. And in my work, we've been showing that subliminal stimuli fail in a number of ways, that you have to be conscious of information in order to reflect upon it for a long time, to discretize the input into symbols, to route this information, and especially, as I will show you, to monitor our behavior and diagnose our errors. So I'm looking here at a particular phenomenon which is called the error-related negativity, which is a wonderful self-monitoring by the brain. The error-related negativity you get when you ask a subject to categorize stimuli, typically using two buttons. In my case, I flash a digit. I ask subjects, is it larger or smaller than 5? Everybody knows the correct answer, right? If it's larger, you press right, and if it's smaller, you press left. You know what you should do. But sometimes you make errors because you're responding too fast. When you do, you get this extremely sharp response of the brain over the anterior cingulate cortex, which is only occurring when you have an error, even in the absence of feedback. It's been called the error-related negativity. A classic but non-scientific name for it is the oh shit effect, because you realize immediately that you should have done otherwise, but it's too late. Okay. Well, it's a very interesting case. How does the brain self-monitor, generate the error in the first place, corrects it? Or at least detects it. In order to study the relation to consciousness, we used our masking paradigm, flash the digit, masked it, forced subjects to respond quite fast, decide whether the digit is larger or smaller than 5, and then tell us whether they were conscious of the digit or not. And the findings have been extremely clear. You get this error-related negativity only on trials where the subjects report consciousness of the digit. This is true at all durations of presentation. So even for the very same physical stimulus, like at 33 milliseconds or at 50 milliseconds, we have the same physical stimulus, but you get the error-related negativity only when subjects report seeing the stimulus. When they report not seeing, they can do better than chance. They click the right button better than chance, but they don't know whether they were correct or whether they made an error. They don't know on this specific trial with the error negativity. Now, we know that the error-related negativity relates to the anterior cingulate and posterior cingulate cortex. Why is it the case that it depends on consciousness? Well, I think this is a case of an all or none type of operation that requires this high-level assembly in the brain. What we think is happening is the following. Non-consciously, your brain is able to perceive a stimulus, propagate the activation to the motor cortex, and therefore generate a better than chance response without consciousness, and even get an unconscious hunch about the confidence in this response. But this is not enough to know on a single trial basis whether you made an error or not. But there is a second route to motor action, which is a conscious route, which is the development of an actual following of the instruction, compare the numbers to 5, generation of an intention to act, a conscious intention to act, and of course, normally you'd like your actions to be guided by your intentions, but in this situation where you're asked to respond fast, sometimes your actions come faster than your actual consciousness of what you should do. In this case, you might do an error, and you can also detect your error by comparing the intention and the action, knowing that if they are different, you probably made an error. So we think this is how the RN works, and we think that the top route is only available when you're conscious. The bottom route is available whether or not you're conscious. So only when you're conscious can you do this comparison and detect that you've made an error. This is one case of an operation that's after conscious ignition. So in order to test this model, we used our decoding. The predictions are very simple. We should be able to decode elements of the bottom route without consciousness, but we should only be able to detect the top activation, conscious intention, and error detection on conscious trials. So this is what we did. We built decoders using the very same data, decoders that are able to use the brain recordings and decide, for instance, what was the stimulus. In this case, I hope you can see this, we can decode what was the position of the stimulus. Was it on the top or on the bottom of screen, even if subjects are unconscious of the stimulus. So this is one case of decoding an unconscious perception. You can see that we decode a little bit better when it's conscious, but we can decode way above chance even when it's unconscious. Now, we can also decode the response that the subject is making, and of course we can again do that on conscious and on unconscious trials. And notice that it's slightly later in time, so these decoders really are decoding different time moments. This is early and this is later. But a little bit later, we can build a decoder for what the subject should have responded. And this is really interesting. The brain is responding in one way, but some brain areas which we can decode are able to say this is not the correct response. And because we have so many errors, we can build orthogonal decoders for the actual response and for what the response should have been. But what you can see is that the brain contains a representation of the intention a little bit later than the action, only on trials where the subject reported being conscious of the stimulus. That makes sense, right? You have to be conscious of the digit in order to know what you should have been doing. Okay. But it's really quite amazing that we can decode the intention independently of the response. It's the very same brain that contains multiple representations. An unconscious representation of the urge to act, and another representation of the actual intention, what I should be doing. And like we expected, we can also decode the errors that the subject is making. So we know from looking at the brain whether the subject is making an error or not, but the brain knows it only when the stimulus was seen. And we can also show that this response is dependent on the strengths of the two others. It looks like a comparison of the two others. So here's one case where consciousness is very efficient. Because we have a stable representation in the brain, we can do more computations than we could if we were not conscious. Now, consciousness during dual tasking. What I've shown you so far is the progression of a stimulus. It's progressing through these assemblies. At some point, it's strong enough to create a conscious representation, or it's decaying. I want to show you another example where actually the contrast between conscious and unconscious is even closer, because the stimulus that I'm going to show now, this stimulus could have been conscious. It's strong enough, but we're going to make it unconscious by having it compete with another stimulus that's already being conscious. So this is a dual tasking situation. We ask the subject to perform 2 tasks at once, and we're going to see that the first stimulus delays or annihilates the consciousness of the second. So the prediction is very simple. Either there should be a strong delay in accessing consciousness, or there should be a complete collapse. The first phenomenon is known as the psychological refractory period. The second is known as the attentional blink. And our work suggests that the 2 phenomena are very tightly related. So these are the predictions of the model. For the first task that you asked to do, you should be perfectly normal. You should have your normal perceptual stages, and then you should have your global ignition. Prefrontal area should show this pattern. But for the second stimulus, according to the model, you should still have your parallel processing of the modality-specific perceptual processes, but then there should be either completely delayed ignition waiting for the first process to complete, or there should be a complete collapse with the inability to retrieve what was the second stimulus and therefore inability to become conscious of it. So basically, this is what we found in a series of experiments. I'm only going to show you the most recent one, which was just published in the journal Neuron a few months ago by Sébastien Marty. We present to subjects a stream of letters, and at the same time, we distract them with a sound that they have to judge whether it's high or low in pitch. For a short while after this pitch, we can show that subjects become perturbed in their letter processing. We vary the lag between the sound and the letter to vary the amount of interference. And here are the behavioral results. First of all, subjects are delayed in responding to T2. You can see the response to T1 here is completely unaffected by the delay with respect between T1 and T2, the lag. But T2 is getting way slower. This is measured in hundreds of milliseconds. You cannot react simultaneously to 2 stimuli in this situation. The second one is delayed. But what we also see is that the second one sometimes is not responded at all. This is the percentage of responding to T2, and you can see that there are actually many trials in which T2 is not properly reported by the subjects. They simply don't detect it, unable to detect whether there was a particular letter that was present in the display. It's called the attentional blink. What's interesting in this situation, we get both phenomena in the same trials, depending on stochastic aspects of brain activity. So what is brain activity like? Just to show you how sensitive MEG is these days, even on individual sensors, we can see that there are some that respond to particular stimuli, such as the auditory stimulus here, or all of the visual stimuli here, or a specific event like the response of the subject. So it's very easy using this machine to decompose brain activity. And again, we use our machine learning technique to do that. So what we do here is we look at task 1 processing. We create a decoder that tells us whether the subject is doing the task 1 or is not doing the task 1. We train it at the longer lags when there is no interference, so there is a long delay between the first task and the second task. We can see this diagonal pattern, which indicates a series of stages of processing the first task. And what you can see on the right here is that when we generalize to the other conditions, we can still tell whether the subject is doing the task 1. We can see that there is very, very little interference with the presence of task 2. So task 1 is essentially unfolding normally. A little bit faster maybe towards the end. But what about task 2? So now we create a decoder for whether the subject was seeing a target letter or not a target letter. This is essentially telling us whether the subject is doing task 2 or not. We train the decoder on the long lags, we generalize to the other lags, and what do we see? What we see is a complete collapse of all of the late activities, just a little bit of activity here, when the subject reports not seeing the letter. So not seeing is associated with the loss of these late stages of activity again. Now, reporting the letter correctly in these situations of shorter lags is associated with the same sort of activation we see in the longer lags, but you can see that it loses the diagonal. It leaves the diagonal and moves to the right, which means a delay. We find the same stages, but they are delayed in time. Let me show this to you closer up, and I think we'll see more clearly what's going on. If we look at each of these different stages of processing identified by our decoder, the early decoder, around 200 milliseconds here, is able to decode essentially constant level of activation, just slightly reduced under all conditions of lag. So whether or not there is interference, as I told you, early processes operate in parallel with each other. The subject may be occupied with the sound, but these stages are unconscious, automatic. They proceed in parallel with the processing of the sound, and they detect that there was a target letter. It's very interesting that the brain is able to see that there is a target unconsciously and using early stages. So even in the blink case, the brain detects the target. If we look at the later stages, by around 300 milliseconds, we see a complete collapse of the black curve here. It returns almost to zero here. What that means is that on blink trials, there is a sort of failure of ignition. The activity drops back to a very low level, indicating no ability to sustain the stimulus, and this will be associated a few hundreds of milliseconds later with the subjects reporting, there was no letter, I didn't see a letter, there was no target for me. If the subject reports seeing the letter, what we see is a dilution and delay of the activation. I hope you can see this clearly. Depending on the stages, this one is a little bit diluted, so it starts at the right time, but the amplitude is much lower and it lasts longer, and all of these late stages are just delayed, like a rigid delay. We can see it better in this representation, which is like a complete radiography of the tasks that are occurring at the same time in a subject. So the blue task is the auditory task, the red is the visual task, and you can see that at a long lag they don't collide. As we make the tasks closer in time, the two targets are now only 400 milliseconds separated here. You can see that we begin to see a little bit of a repulsion effect. But as we go to 100 millisecond delay here, 200 or 100 millisecond delay, you can see what's happening. The first task is essentially unfolding normally. That's the blue task. The second task is starting normally. This is this diagonal pattern here, overlapping with the blue. And then suddenly you see this rigid delay, and everything else is delayed in time. Okay. So there is a bottleneck in the system, and that bottleneck coincides with the reportability of the stimulus by the subject. Subjects report the stimulus only when there was this ignition phenomenon, the stimulus crossed the threshold. But even when they're able to report, they have delayed activation. Now, this delayed activation is very interesting, but how does it relate to consciousness? How do we know that it has something to do with consciousness? If my view is right, during this dual task situation, subjects are not only delayed, they are unconscious. They are not even conscious that there was a T2 stimulus. They can only be conscious of the second stimulus when they had this ignition pattern, so late in time, okay? So how do we know that subjects are blind to the second target stimulus waiting? Well, we did an experiment with Sébastien Marty some years ago. We present this stimulus, auditory stimulus, and just now a single visual stimulus, so they see them all the time. And we ask them to report their subjective experience of the trial using many subjective questions. This is a very interesting method. We can ask about subjective reaction time. How long do you think it took you to respond? And subjects are very accurate and highly correlated with the objective reaction time for the first task. Okay, we now also ask them questions such like, what do you think was the delay between the first stimulus and the second? Do you think there was a short delay or long delay? We can be very quantitative. We train them to report using this scale here. And this is a summary slide of what objectively happened. The reaction time 1 here on average, and the reaction time 2 as a function of the lag between the two stimuli. You can see that when this is stimulus onset asynchrony, SOA, this is the same thing as the lag. You can see that when the lag is very short, subjects are super slow in responding to the stimulus. When the two are simultaneous, the second one has to wait a long time. And then as we make the second later and later, you can see that the reaction time becomes essentially normal and short. Well, this is the subject's introspection of that stimulus, reconstructed from all of these subjective questions we ask them. Subjects have very good introspection of the first task, and even on a trial-by-trial basis, which I'm not showing you here. They also have some introspection of the second task when it's late in time, but they have absolutely zero introspection of when the target stimulus appeared and how long it took them, how long it was waiting, basically. So what you see here is that subjectively, the reaction time 2 does not appear to vary with the SOA. Subjects think that they always took the same time to respond. They don't realize that they've been taking a long time to respond to the second stimulus. They also don't realize that the onset of the second stimulus varied. They think that it was always coming when they were finished with task 1. This is the most amazing situation. The stimuli are simultaneous, objectively speaking, but subjects think that the second stimulus came way later, only when essentially when their workspace system was free to process that second stimulus. So we call this the blind spot of consciousness. It means that inattention, in this case, temporary inattention breeds unconsciousness, either permanently, this is the attentional blink, or temporarily, when you're delayed like this, temporarily you have a blind spot of consciousness. This is the sort of dual aspect of dual task processing. First, you are slow, but second, you're not even conscious that you are slow. You're not conscious that you're not conscious. Sounds very trivial that you're not conscious, but I think this has deep consequences because it makes us do things like drive with a cell phone, right? Because we are going to be slower if there is a need to react consciously, but because we're not aware that we are slow, we still do it, right? We're not aware that we're not aware. Okay, last part of my talk. All I've told you is using external stimuli. We're bombarding the subject with stimuli and we're asking whether they become conscious or not. But if the view is correct, we shouldn't have to do that. The workspace system is autonomous. It has autonomous activity and we should be able to see signatures of consciousness in brain activity. So I'm starting this movie here, which is the classic word of Mark Reichel and his collaborators, showing that there is spontaneous activity in the brain. And I always was intrigued by this spontaneous activity. The brain is constantly traversed by structured activity, forming global patterns. And they even sort of predict the content of consciousness. So using the particular patterns that you measure, you might be able to tell whether the subject is mind-wandering or whether he's using mental imagery. When this pattern occurs spontaneously during sleep, you might even predict if the subject wakes up what sort of dream he was having. So there is beautiful work correlating this pattern, at least partially, with subjective contents of consciousness. And one might wonder whether this is just William James' stream of consciousness. It really looks like unfolding patterns of global brain activity, workspace activity patterns. But there is a problem. Paradoxically, this sort of long-distance activity seems to occur still when the subject is no longer conscious, for instance, under anesthesia. And this was first shown by Vincent and collaborator in Max Reichel lab, where they showed that you can have these long-distance functional connectivity patterns even during anesthesia. So what's going on here? I always thought it was a little bit weird, but I always thought it was in the dynamics that the solution was. If my view is correct, there should be constant changes in this functional connectivity pattern that contain a signature of consciousness. The dynamics should contain a signature of consciousness. So here's what we did. This is work that was published just a year ago by Pablo Bartfeld and collaborators. We analyzed resting state data from either awake monkeys in the lab or monkeys that we anesthetized with propofol, either moderate or high doses of propofol. But this was general anesthesia, and that's why we did it in monkeys, by the way. But the way we analyzed the data was different from usual. We analyzed it over time windows, so that we had a sliding time window, as you can see here, and we could compute functional connectivity matrices in specific time windows. And you immediately see the result here. The functional connectivity is not stable during the awake state. It's constantly changing. And if we analyze it in this way, first we replicate the presence of long-distance connectivity, functional connectivity, in the anesthetized monkey, just like Vincent and collaborators and many other groups since then. But we also find that there is dynamics to this functional connectivity, and we can find many states of functional connectivity. We can decompose these dynamics into many superimposed states, and we find that these states are more diverse under conscious conditions and under unconscious conditions. So what you see here is the awake animal has a high probability of being in any of these 7 different states, whereas when the animal is anesthetized, you see that some states begin to dominate and some disappear completely, and the diversity is lost. Now, what's more interesting, I think, is we can predict which states are staying and which states are disappearing during anesthesia. The states which are staying are the ones that are closest to the anatomy of the animal. So we have the anatomical connectivity matrix. This is approximate. It's called the COCOMAC. It's a essentially database of connectivity of the macaque. We can do better these days, but this is good enough for this analysis. The states have been sorted by their proximity to the anatomy. And what we can see is that during anesthesia, the states that stay are the ones that are closest to the anatomy. So what's going on here? Well, we think it's very simple. During anesthesia, of course, there is still brain activity. The brain is not completely silent. And this activity is shaped by the anatomical networks that, of course, are still preserved. The anatomy has not disappeared. So what we see is probably random spikes going through this anatomical network, still going through loops, and therefore tracing the anatomical network. And that's why functional connectivity still exists in the anesthetized animal and is dominated by patterns that look like anatomy. But when we are conscious, we have a much greater diversity of functional connectivity patterns that depart more from the anatomical networks. And these are all these networks on the left here. You can see that in these networks, there is a part which is red, which is positive, and a part which is blue, which is negative. That means we select a subset of connections that we're going to use positively, and we inhibit the rest. We select a subset of active networks during the conscious state. And this is a signature of consciousness. We're now finding it not just with propofol, but with other anesthetics and also during sleep. It seems to be holding up as a dynamic signature of consciousness, measurable even with the slow dynamics of fMRI. So a conscious brain is one that has a series of ignition patterns, either evoked by the external world or evoked internally by our internal goals, but still there is this sequence of global brain states that we can measure. I'm just going to close here by mentioning that there is validation of these ideas from all sorts of directions. One of them is clinical. We are working with Lionel Nakache on patients with loss of consciousness, disorders of consciousness, coma, vegetative state, minimal consciousness, and the same signatures seem to be holding up to a large extent. So first we can present external stimuli. In this case, we use auditory stimuli, and we find that there are some patterns of brain activity that are metastable, creating this sort of square pattern of generalization. I won't go into any of the details. This is what we call the local global test in collaboration with Tristan Beckenstein. We're starting this. And if we find this metastable pattern of activity, it's very likely that the patient is conscious or is going to recover consciousness in the next few days. It's a good signature of consciousness. Second, we can also rely on spontaneous activity, ongoing fluctuations in brain activity. And here again, we derive a measure, this time from EEG, of synchrony of long-distance brain networks. We use a measure of sharing of information, long-distance information sharing. I won't go into any of the details, but the strength of information sharing is reflected here in the strength and color of these connections, and you can readily see that the patients who are still vegetative seem to have much lower information sharing over long distances of the brain than the patients who are conscious, and several similar signatures. So I think this is where we are today. We have, in fact, a wealth of signatures of consciousness. This should not be surprising, because if this workspace idea is correct, becoming conscious of a piece of information is really broadcasting it at the brain scale, and this is a very intense event. It's recruiting a lot of distant brain areas that work together, so it's not something which is difficult to measure. It gets reflected in a lot of events. At the same time, there's still a lot of unconscious parallel processing going on in the same brain, and we've learned to separate it more and more. I'm leaving you with the people who did the work, Lionel Nakache, who works with the patient, and all of the other guys here working, helping me to do the decoding of these brain signals. And I just want to mention that I synthesized all of these findings recently in this book, Consciousness and the Brain. Thank you very much for your attention.
Thank you very much. In the interest of time, we're going to have to move to our next speaker, so hold your questions until the panel at the end. And I would like to invite... Oh, okay. How long will it... Well, let me introduce Anil in the meantime. So it's my great pleasure to introduce Dr. Anil Seth. And I'm particularly proud because despite his long-standing and highly contributory career to the science of consciousness, this is the first time that Dr. Seth has attended the Tucson conference, so I'm excited that he joined us today. He is a professor of cognitive and computational neuroscience at the University of Sussex, where he is also the co-director of the Sackler Center for Consciousness Science. He also serves as the editor-in-chief of the Neuroscience of Consciousness, which is the official journal of the ASSC, And so please join me in welcoming him.
Thanks very much. It's indeed a great pleasure to be here. I was going to say it myself, I've been involved in this field for a long time, but this is actually the first time I've been to towards the science of consciousness, but now it's the Science of Consciousness meeting, so I'm very excited to see what the rest of the week brings. And what I want to do today is, following the organization of these two plenary sessions, is to give you a tour of one approach. And this is not the only approach that I think holds promise, but it's an approach that I'm particularly excited about at the moment, and it's this idea of predictive processing, or the Bayesian brain, and how this might help us understand the neural mechanisms that shape conscious phenomenology. So let's see. Oh, just got to put this thing in here. So this is where I come from. The Sackler Center for Consciousness Science was founded about 10 years ago. Does this? Oh, yeah, that's nice. Founded in 2010 with myself and Hugo Critchley as co-directors. And what we try to do with this center is take a largely cognitive and computational clinical perspective on consciousness, try to unravel the biological mechanisms that underlie conscious experience, with a view to informing clinical treatments, mainly in psychiatry but also in neurology. The idea that psychiatric conditions are often not well understood at the level of the mechanisms that give rise to the specific kinds of conscious experience disturbances that we see. And of course, on the flip side, you can take populations of psychiatric populations and study them to gain insights into what we might take for granted in our daily conscious lives. And of course, there is also this journal which has been going for a couple of years now with Oxford University Press. Just want to mention about that it's an open access journal, which usually means you've got to pay to publish. But because we've just started and getting going, it's free for the rest of this year. So if you've got incredible data to publish about the neuroscience of consciousness, submit it before the end of this year, and you won't have to pay any charges, which is great. Free. So when thinking about consciousness, I like to think of three different dimensions. I like to think of conscious level, which is the difference between being unconscious, as in general anesthesia or coma, and fully conscious, awake, alert, and aware, as we are all now. Then there's conscious content, which is when you are conscious, you're having particular phenomenal experience. There are percepts, thoughts, beliefs, emotions, moods, these elements of your conscious scene that are present whenever you are conscious. And we can ask, what underlies being conscious of something when you are conscious? And one of the things you're conscious of when you are conscious is the experience of being you, the experience of being a self. And this has many levels too. There's very basic levels of the experience of identifying with or owning a particular body, of being a body, all the way to the experience of being an individual with a name that has some continuity over hours, days, months, and years. And it's easy perhaps to take this experience of selfhood for granted because it's somehow always there in the background. But I will show you, I hope, that examining the neural mechanisms of self-experience is particularly interesting. So today, I'm not going to talk about conscious level at all, even though we do work on this in the lab. And I'm going to focus on conscious content and conscious self, and try to give you a bit more of a high-level, engaging overview without getting too much caught up into data. I'll give you some data as well. So conscious content. The hero here is a chap called Hermann von Helmholtz. He was a German physiologist in the 19th century, and among his many contributions, he was the first to discover the speed of conduction of electrical impulses along nerve fibers. He also came up with the idea of perception as unconscious inference, and the idea of this is really very simple, that sensory signals come without labels. They don't even tell you which modality they're in. They're noisy and ambiguous with respect to the objects in the world from which they arise. And if the purpose of perception is to make sense of sensory data, to know what's out there in the world, there has to be a process of inference in which the brain brings prior beliefs or expectations about the origin of sensory signals to bear on making sense of this noisy and ambiguous data. And he called it unconscious inference because we're not necessarily aware of the process of this inferential process, only the results. So that's the sort of basic idea. William James, since we're talking about consciousness, I've got to tell you that, of course, William James said exactly the same thing. He said, whilst part of what we perceive comes through our senses from the objects before us, another part, and it may be the larger part, always comes out of our own head. A number of other people I want to just throw up here who've influenced the way I've been thinking about predictive processing in recent years. This is in rough temporal order: Richard Gregory, Chris Frith, Carl Frist, and Jakob Hochwi, who we'll be hearing from the end of this session, which is to say in a couple of days, and the later Andy Clark, have all written, I think, very interesting and influential things about predictive processing. What I'll say now, I'll repeat it a bit later, is that predictive processing is not a theory of consciousness. It's a theory of how brains might work in general. And you can think, why am I here talking about it then? You can think it's a bad thing, or you can equally think it's a very good thing because it gives us a general framework within which we can ask more specific questions about the mechanisms shaping consciousness. It's rather easy to show that our perception is somehow inferential. And I'll just use one demo here. If you look at this, I don't know if you've seen this before, but if you haven't seen this image before, it's one of a class of image called Mooney images, which is a photo that's been, if you look at this and you haven't seen it, you will probably just see a load of black and white splotches. Hopefully that's what you see. And now, if I just fill this image in, you'll see it's not just a jumble of black and white splotches. There's an image there. There are objects there. There's a woman kissing a horse, for instance. Kind of obvious thing that you might choose as an image. So if you look at that for a while, I won't leave it up there for too long because I don't have time, but if you look at that for a while, and if I now take it away, unfill the image, then hopefully you should still be able to see the objects. You still perceive this image as partitioned into a horse and a woman. Of course, the sensory data is absolutely identical to what you saw the first time. The only thing that's changed is your brain's expectations or prior beliefs about the causes of that sensory data. And so that example is enough to show that our brain's expectations change what we perceive independently of the sensory data that it's given. And there's many other examples like that. The formal framework for thinking about this is long established, and it's very simple, and it's, of course, Bayesian inference. And there's a very, this is just a few curves that illustrate what Bayesian inference is all about. In the context of perception, we can think about sense data being a sort of probability distribution. That is to say, maybe along this axis, there's the sensory data that you get is sort of around here. That's the bit's probability, peaks around here. That's the job of the sensory system. But the brain might also have some prior beliefs. So an example of this might be if you see a face and it looks like your friend and it's outside on a sunny day like today. The sensory data is very, very clear, so it has a lot of weight. Your prior belief that your friend isn't here, maybe you didn't think he or she came to this conference, is going to be overridden because you've got very clear sensory data. You think, oh yeah, my friend's here. And so your percept, the brain settles on what's called the posterior distribution, which is the sort of combination of these two distributions, the optimal combination. And in this case, it sort of comes in sort of in the middle. Now, if the sensory data was less clear, like it's not a sunny Arizona day, but it's like back in Brighton where it's probably raining and it's misty and cloudy, then your sensory data is going to be more ambiguous, so your posterior would be drawn more towards your prior, that it's not your friend, so your percept might settle into something quite different. Of course, this framework of Bayesian inference is not just something that was developed to try to explain perception. It's a very general framework in probability theory that's been used to do all sorts of things like break codes in Second World War or find missing nuclear submarines and do medical diagnosis, more importantly. When thinking about the brain this way, we immediately sort of come into conflicts with some quite classical views of how perception works. And this is a bit of a straw man, but the classical view of perception is as a sort of bottom-up or feed-forward process where sensory signals come in to the eyes, and then they get processed percolate through the brain to ever higher and higher areas. And at each stage, the representations or the things that areas respond to become more complex, starting with just maybe luminance patterns, and as you get deeper into edges, textures, and movement, and then objects and people and whole scenes. But perception in this view is thought of as a bottom-up feed-forward process. The predictive processing view kind of turns this on its head and says that the heavy lifting done during perception is not so much about the sensory information that flows in, but it's more about what flows out. So rather than thinking about feed-forward or feedback, I prefer to think about inside out and outside in. So you have sensory signals that come from the outside in, but you also have these signals that come from the inside out that carry or convey the brain's hypotheses or predictions about the causes of sensory signals. And we've known that there's a great preponderance of these inside-out or top-down connections in the brain. And the predictive processing story gives us a functional interpretation of what these connections might be doing. We can get a little bit more formal about this by mapping this process of feed-forward and feedback, or inside-out and outside-in, onto how cortical layers are organized. So here's a schema of just 3 cortical regions. And the idea is that predictions, the brain's hypotheses about what's going on, flow in this inside-out direction from deep layers of each region to superficial layers of a region below. And that prediction errors flow in the other direction, also with a particular laminar organization. The take-home from this is that what we tend to think of sensory information coming in is really just prediction error. It's just the discrepancy between what the brain expects there to be and what it encounters at every level of processing, whether it's right at the sensory surfaces or further into the brain. At the bottom here is this important feature of precision weighting. And this is just this idea that the brain always has to balance how important the sensory information is with its prior beliefs. And this goes back to that example of seeing a face on a misty day. If you expect the sensory data to be noisy and imprecise, you'll give more weight to your prior beliefs. And the process of attention is thought to reflect this process of precision weighting in predictive inference here. So as I said, predictive processing is not itself a theory of consciousness. It's a theory in general of how the brain does not only perception, but also action and cognition. But it does, I argue, provide a very powerful framework, because it's not fundamentally about consciousness, for asking questions, for exposing the processes that shape conscious experience. And I want to give you a couple of examples of how that story goes. Start with some very old data. I mean, it's been known for a long time that the integrity of these inside-out, top-down connections is very important for conscious awareness. This is some old work by this paper by Alvaro Pascaleone and Vince Walsh. And what they did was they used transcranial magnetic stimulation, or TMS, to selectively knock out the top-down, or I'm just going to say top-down, it's quicker than inside-out, the top-down signals when somebody was doing a visual perception task, in this case, categorizing visual motion. And what they found was that knocking out the top-down signals preserved their objective ability to do the task, but knocked out their subjective conscious awareness. And that was done many years ago. Much more recently, and this is work not from my lab, but from Lars Muckli in Glasgow, using a kind of decoding algorithm similar to what Stan was just talking about, found that you could decode contextual information, so not specific information, but a sort of what a visual scene was like, the sort of thing that might be conveyed by a prediction. You could decode that from superficial layers of visual cortex, even when there's no actual sensory input to those areas. So this is good evidence that these top layers of cortex are receiving information, predictive information from layers in different places. Now, I want to give you a couple of examples from our own lab about how we can ask what within this story of predictive processing corresponds to what we're aware of. Are we aware of the predictions? Are we aware of the prediction errors? At what level? And so on. There's a lot of questions you can ask. Perhaps the most basic question is, do we see what we expect to see, or do we see what violates our expectations? And this is work with this particular study with Yair Pinto, and this is a technique called continuous flash suppression, where we just It's a bit like binocular rivalry. There's a face which gradually appears out of a noisy blur of colored squares. And the subject is led to expect there to be a face or to expect there to be something else like a house. And the short story is, if they expect face, they see face more quickly. It breaks through into consciousness faster. It's as if once your perceptual predictions are validated, then that reaches your conscious awareness more rapidly. Another similar story, and I won't go into the details, can be told with another paradigm called motion-induced blindness. This is the sort of thing where if you stare at the center, that red dot will occasionally disappear. It won't here because it's not ideal conditions. And depending on your expectations about what happens to that dot, it will reappear more quickly, again, if your expectations are valid. The experiment I want to focus on a bit more is, and this is the only kind of low-level psychophysics stuff I'll talk about, has all to do with this fellow. And this is a Gabor patch, and a lot of people in cognitive neuroscience spend their lives thinking about Gabor patches, which are just these boring gratings. And the world is not full of them, but they're very, very good at isolating what happens in the visual system. And a very, very simple task for someone is to just say whether a patch was present or not on a given trial. This is work with my PhD student, Maxine Sherman, and also Ryota Kanai. We were both the supervisors, and she's just finishing her PhD now. This is the paradigm, and I won't belabor the details. Basically, people are asked to detect whether one of those Gabor patches was there or not. And in a given block of trials, they're either expecting the patch to be there, it's going to be there 3/4 of the time, or they're expecting the patch to not be there. So we can manipulate their prior beliefs about what will be there in the world. These two rows, basically here, people are also attending to the task, and here people are doing something else in a dual task paradigm, again, similar to what Stan just mentioned. The only point of this is because we want to dissociate the role of expectations from the role of attention, because they're often conflated. Now, people make a forced choice response, that is, they say they're forced to guess whether the patch was there, but they also give a subjective confidence rating. And the argument here is that when they say they're not guessing, when they're sure about what they say, well, that aligns with their conscious experience. Whereas if they say they were guessing, then they probably weren't conscious of anything that underlined their decision. So that's sort of roughly subjective perception. It also means we can bring to bear the machinery of signal detection theory to now try to quantitatively separate how people are doing objectively and how people are doing subjectively. So the idea, very simply here, is that we want to quantify the degree to which people's confidence tracks their accuracy. Do they know that their perceptual decisions were correct or not? If they do, if they're good at that, then we can infer loosely that, well, that corresponds to them having been conscious of the stimulus. So the first thing we find is that if people make a response that is in line with their expectations, they're firstly more confident, and their metacognitive sensitivity is better. This is just behavioral stuff. What does it mean? It means that if what you do is in line with your expectations, it's more likely to be part of your conscious percept than if you make a response that's not in line with your expectations. So expectations in this case are influencing whether you're subjectively aware of a particular perceptual decision. We also wanted to look about the underlying brain activity a bit, and here I just focus on one example, and this is to do with the alpha rhythm. Now, we know that the brain is oscillating at various frequencies all the time, and especially over visual cortex, the alpha rhythm at about 10 hertz is particularly prominent. What does it do? Well, a lot of people have said it's just the brain idling, it's doing nothing, it's waiting for some input, because when you open your eyes, the alpha rhythm is suppressed a bit. But perhaps it's doing something more interesting. And the specific question we asked was that, do your expectations about the world come into play at particular phases of the alpha rhythm? The thought is that this oscillation might orchestrate the exchange of predictions and prediction errors in a sort of iterative process. That's the hypothesis. There's already quite good evidence from a number of studies that pre-stimulus alpha phase, that is where the alpha cycle is in time, can affect a lot of perceptual processes like attention, decision-making, and so on. And here we wanted to ask the same question about expectation. So what Maxine did, and this is again with Ryota and also Rufen van Rillen in Toulouse, was to ask whether there was a particular alpha phase or a particular time point, prior to stimulus onset, where alpha phase predicted whether people would say yes or no. That's the first thing. And we find there is indeed, in the alpha range at about 100 milliseconds, before the stimulus appears, that already predicts whether you're likely to say yes or no. And then using that that period there, that part of the algorithm there, we can bin all the trials into whether people were successful, whether they missed a target, whether they made a false alarm, so that we can apply signal detection theory. And the short story of all this is that by doing that, we can quantify to what extent people's decisions are, people's expectations are affecting their decision, and does that fall at a particular phase of the alpha cycle? And we find that it does. There's a sort of optimal phase here in the pre-stimulus alpha, so the ongoing brain rhythm, at which your perceptual expectations have most effect. That means there's an opposite phase where the sensory data has most effect on what you perceive. They're out of phase. And this doesn't prove our story, but it's kind of in line with the idea that this algorithm is indeed orchestrating this interchange of perceptual predictions and prediction errors. And the same thing is true a little bit for confidence. So this is an intriguing possibility, that alpha oscillations orchestrate this business. And we've done some other work which I don't have time to talk about, which is also consistent with this idea. There's this interesting phenomenon called perceptual echo, in which if you just show a fast-changing luminance pattern, then the brain actually reverberates along with that luminance pattern at about 10 hertz as well. It's not the alpha oscillation itself, there's this perceptual echo or reverberation. And we find that this reverberation can learn to predict these fast-changing temporal dynamics. So it's, again, implicating this oscillation in predicting what's happening next in the visual system. So that's all very well. Now I want to move on to tomatoes, because, as I said, we don't actually go around the world perceiving Gabor patches. Our perceptual experience is much richer. And one of the, I think, the interesting opportunities of the predictive processing story is we can start to ask questions that are more directly relevant to our everyday perceptual phenomenology. One aspect of this is that objects mainly seem to be real. When I look at that loudspeaker, I know it's not a picture of a loudspeaker. It has a certain perceptual objecthood, reality, or presence to it. And how does that come about? Whereas if you look at this, you know it's not a real tomato. It's an image of a tomato. What explains that difference? There's quite an old psychological theory called sensory motor theory, which explains perceptual presence as resulting from an implicit mastery or knowledge of the sensory motor contingencies that govern how sensory signals respond to actions. Put very simply, this means that what they're saying is that somehow my brain is encoding or predicting what the sensory signals would be were I to make a particular action, move my head in a particular way, or pick an object up and move it. And that if there's a rich sort of encoding of these conditional relationships, then I will experience that object as having some objecthood or perceptual presence. But they didn't provide any mechanism for that. But predictive processing can start to put some mechanistic flesh on those bones. A lot of the people who sort of originated sensory motor theory don't like that because they intended that theory to be free of any kind of internal models, whereas predictive coding is rich with internal models. It's basically saying that's how we do perception. But if we equip predictive perception with action now, you can see there are two ways we can reduce perceptual prediction error. We can either update our predictions about what's there, or we can make actions to change the sensory data we get. And this is called active inference, and it's a concept elaborated primarily by Carl Friston. But if we're going to make actions to reduce prediction error, then our brain needs to know what is likely to happen to sensory data for any particular action, that it has to encode something about these conditional relationships between a particular action and the sensory data it expects. So there's this sort of counterfactual aspect to predictive processing that we can build in. And the idea simply is that we now see objects as real when the brain's predictive models encode a rich repertoire of these conditional counterfactual relationships. There's a very long paper I wrote a while ago which goes into extensive and boring detail about all this, so I won't. But one of the things I tried doing that is to account for a funny thing about synesthesia. Now, you all know about synesthesia. This is one example of synesthesia where letters appear to, for synesthetes, black letters will also have a concurrent experience of color of some sort. They may be experienced as red or green or whatnot. But in synesthesia, it's almost universal that synesthetes know and do not experience those colors as real. They don't experience them as existing in the world. They know that the letters are still black. So why might that be? Well, one reason that might be is that there are no conditional counterfactual relations between the predictive models that are involved. Unlike a real color, a synesthetic color does not change as I move my head around it. It doesn't change. So it may lack that kind of perceptual reality. Now that's all very well, but you might think all these ideas are a bit highfalutin and a bit hard to test. One of the things I'm quite excited about now is the potential of technologies like virtual reality to start to put more conceptual theories like this to experimental test. And this is one example of how we're testing this idea of counterfactual predictive processing and objecthood. We use augmented reality here to introduce a series of virtual objects, and then we can use the VR software to change how these virtual objects respond to your actions. So here's an object that behaves normally as you move your head around it and twizzle it around and so on. But we can also have objects, one will come up in a second, I hope, which have other and slightly strange behaviors. This is like the moon. It always presents the same face to you no matter what you do, which is a very unnerving and slightly perceptually uncanny experience. And there's another one where we can make the sensory motor contingencies random. And of course, now we can start to ask questions about whether people experience these virtual objects as real in various ways, and that's one of the things we're doing. I will summarize this first part on conscious content. Conscious perception reflects the brain's best guess of the causes of sensory signals. Perception, in this case, is like a controlled hallucination, or hallucination is like an uncontrolled perception. And there's a lot of data, accumulating data now showing how expectations can accelerate conscious access, can enhance confidence and metacognition, so they have to do with the conscious part of perception, and they have definable neural mechanisms in all sorts of ways. But then zooming out, we can start to ask interesting questions about how within the predictive coding framework, we can start to understand things like hallucination and presence and more interesting aspects of real-world phenomenology. So the first take-home is that conscious content is a Bayesian fantasy constrained by reality. The second and last thing I want to talk about is conscious selfhood. Part of what we experience when we experience is being you. It's the fundamental experience of being someone. And this has, as I said at the beginning, many levels. And I just want to focus on one here, which is the bodily self, the experience of being and having a particular body. William James again said our mental life is knit up in our corporeal frames, which is just, I think, Victorian for saying that the body matters. There are two traditions for thinking about how our experience of body ownership arises. One is the perception of the body from the outside. So we see our bodies, and in a sense, proprioception, we perceive our body position and configuration in space. That's sort of the outside. We can feel touch on our body. The other tradition is perception of the body from the inside, and this is all about interoception. A lot of what our brain is doing is responding to and controlling its own internal physiological state. And just as the external world is not known to the brain, the internal world is also not directly revealed to the brain. It also has to make its best guess about what its internal physiology is. So there are these two traditions to thinking about perception of body. And the proposal I've been thinking about for a while now is there's common mechanisms underlying how we experience our body and how we experience the world. That is, selfhood depends on predictive models of the causes of self-related signals across exteroceptive, that's vision and audition, proprioceptive and interoceptive domains. So we have a picture a little bit like this. All these processes going on at once. And at higher levels, we have predictions that integrate these different modalities. So we make joint predictions about interoceptive and proprioceptive and exteroceptive signals. One thing that immediately follows from this is that our interoceptive percepts, how we experience our own internal bodily state, is the brain's best guess of those states too. So we have this process of interoceptive inference where we might have some signals like heartbeat-related signals that are then flowing into the brain to be met by downward-flowing predictions about those signals, perhaps from the anterior insular cortex, who knows? So we have this counterflowing predictions and prediction error signals, this time in the interoceptive domain. Lisa Barrett has taken this idea a bit further by showing that the anatomy of the interoceptive and visceromotor cortex is supports this kind of process of interoceptive predictions. One thing that follows from this is that emotion might be the result of this inferential process on interoceptive data. So just as our external visual perception is about inference of visual signals, emotion might be our perceptual inference about interoceptive signals. But it also plays into how we experience what is and what is not our body. This is the classic rubber hand illusion. I'm sure you've all seen this. That's the way to test it. So in that example, we're seeing how surprisingly malleable and flexible somebody's experience of what is and what is not their body really is. It's surprisingly easy to convince someone that this plastic hand is, in fact, in some way part of their body. And this is done by having simultaneous visual and tactile input while hiding the person's real hand. Now, this can be understood as the brain's best guess as well. It sees something that looks a bit hand-like. It sees it in a position which is kind of where a hand might be. It feels touch. And so the brain thinks, wow, it doesn't look much like my hand, but that's my best guess, that it is somehow my hand. And that works very nicely. So what we wanted to do, again using some VR stuff, is it moving on, is to ask whether the same thing applies between, let's say, visual perception and interoception. So does the brain put together interoceptive signals online with other sensory signals in deciding what is and what isn't part of its body? And we use this cardiac feedback paradigm here, where instead of a rubber hand, we have a virtual reality hand here. So there's Microsoft Kinect, which is making an online model of somebody's hand, and it's pulsing to red and back very gently. And we can make that happen either in time or out of time with the person's heartbeat. And what we find is that when the virtual hand is pulsing in time with somebody's heartbeat, then you're more likely to experience that virtual hand as your own hand. So there is this online integration of interoceptive signals that helps us determine what is part of our body. We can also do other things with this paradigm here. For instance, we can embody you with a virtual hand and have it change color to see if you become more or less racist when you embody a cultural outgroup hand. And my personal favorite is coming up next, which is where we can radically change the size of the virtual limb to make it smaller or larger. So here you suddenly feel you've got a very big hand or a very small hand. And there are real-world conditions like the Alice in Wonderland syndrome, where people experience their body shape as telescoping up and down in size. And of course, in more moderate situations, we have body dysmorphias, where people may have more subtle distortions of their body's shape, which we can now sort of objectively measure using these kinds of paradigms. So that's just the results of that experiment. I probably don't need to go through them. Now, I want to finish with some more speculative stuff. And this is applying how far can we go thinking about predictive processing in terms of our experience of being a body or being a self. So here's this framework where we can minimize prediction error either by updating our predictions or by making an action to change the data that we get. And in the interoceptive world, we now have interoceptive prediction errors. So we can either update our interoceptive predictions, or we can engage in autonomic control. We can change the physiological state of the body to make our interoceptive predictions come true. And this highlights a very interesting distinction between what we can call epistemic active inference. And this is where we make actions to find out more about the world. So when we make eye movements, these are often to discover information about what our visual world is like or what's out there. But there's also instrumental active inference, where we use action to control sensory variables. So we don't care so much about what's out there in the world, but we want to control or regulate that variable which we sense. Even in exteroception, we do this when we're, say, catching a cricket ball. We keep the tangent of the angle to the ball constant so that we will catch it. It's a perceptual control rather than making an action to find out about it. This calls on a very different tradition from the 20th century called cybernetics, and in particular the work of Ross Ashby, which I want to finish with because I think it's quite exciting and relevant. Ashby, one of the founders of cybernetics, treated organisms as having this fundamental imperative to maintain essential physiological variables within viable bounds. So your heart rate can't go too high, your blood pressure has to remain quite well regulated, and many other things 2. If you go out of bounds, then you die. And he had many ideas about how this might happen, but one of his last papers, the clue is in the title. He says, every good regulator of a system must be a model of that system. And this is very much like this instrumental active inference, that if we want to effectively regulate a variable, we have to have a good predictive model of how causes, perturbations might affect that variable. And this is predictive coding used for a very different purpose now. It's for control and regulation. So this is the sort of idea now of the cybernetic Bayesian brain. So whereas exteroception emphasizes finding out about the world, inference to the best explanation, we can think of interoception as emphasizing control, inference to the best prediction, not best explanation. And the best predictions in this case are those which enable control, regulation, and physiological homeostasis under a very broad repertoire of perturbations. So to put it in one quite long-winded sentence, the experience of embodied selfhood, in this view, depends on control-oriented active inference that regulates physiological variables. One thing that's quite nice about this is it's a new way to look at an old debate in cognitive science, which is how mind and life are connected. You know, mind isn't just a computer program running on some physiological hardware. Mind and life are intimately bound together, but how and why? And if we start to think of predictive models implemented by brains being there because they're in the business of regulating physiology, then we can start to understand how mind and life connect. This brings us all the way back to Descartes at the end. Descartes was rather infamous for many things, but one of the things was for claiming that non-humans lacked rationality or consciousness because they were just beast machines. They were just physiological bags of flesh that did what they did. And later, Julien Lemaitre extended this to humans as well and said, well, you know, we're just beast machines too. And the idea was that being a beast machine was in some sense incompatible or an alternative way of thinking about a creature than being a rational, conscious individual. So all this stuff I've talked about is leading me to think rather the opposite, that we are conscious and we have an experience of subjectivity of being a self because we are beast machines, because we are self-sustaining bags of flesh whose brains care about the persistence of the body over time. And this is the only image I could find of beast machine, by the way. There's a Transformers film about it. So to summarize the whole thing then, the experience of selfhood is also a process of best guessing. Embodied selfhood depends on this control-oriented active inference process across many different sensory domains. And predictive perception, then, is a consequence of a more fundamental imperative to maintain homeostasis, to stay alive. In summary, I predict myself, therefore I am. So I just leave you as standard with the people who are working in the lab in Sussex, and I've highlighted in red those whose work I've described. Asa Chang and Yaya Pinto, and Maxine did a lot of the psychophysics stuff, and Kesuke Suzuki is my virtual reality whiz kid there, and Ryota Kanai and Rufen van Rillen have been involved also. Thank you very much. Thanks.
Thank you very much for that fascinating talk. Again, in the interest of time, we're going to move to our next speaker. Because this is Tucson, the man needs no introduction. It's Stuart Hameroff. Before Stuart starts his talk, I just wanted to say that we will continue this session tomorrow where we're going to hear about integrated information theory, some higher order theories, and then the final session will be a consideration of all 5 potential roads to conscious experience. And with that, I will turn it over to Stuart Hameroff.
Thank you, George. No, that's not it. No, I was on the first one.
First slide, or? Let's see here, maybe there's a different.
No. That's it. Oh, thank you. I just use this. Okay. Thank you very much. Thank you, George. Thank you, everyone. So my talk is titled "The Road from Within." This is part of a two-session deal on 5 roads to consciousness, and I'm going to talk about what's going on inside neurons. And what you see in the background of this picture is an immunofluorescence image of a neuron. Everything that the other speakers have talked about occurs at the surface of the neuron on the membrane, and I'm going to talk about what's going on in the cytoplasm. The yellow is immunofluorescence microtubules, and the red is actin. So, of the 5 roads to consciousness, the other 4, global workspace, consciousness correlates with, as we heard from Stan DeHaan, sudden widespread activity in a subset of cortical neurons. Predictive coding from Anil, comparing, updating by feedback using Bayesian probabilities. Higher order thought we'll hear about tomorrow. Feedback from prefrontal executive cortex to other areas. And integrated information, maximal integrated information, or PHI. And all of them are more or less along this scheme. This is actually for higher order thought, in which from the thalamus, for vision anyway, primary inputs go to V1 and then forward in a feed-forward mechanism, and then feedback somewhere else in the brain. Stan talked about two pathways. I'm not sure if he was counting this one, but this is more relevant, specific for higher order thought. And somehow they all have the implication that conscious perception occurs several hundred milliseconds after sensory input. And Bing happens. As you know, I've used Bing in my talks over the years to indicate conscious experience, and in a major breakthrough in consciousness studies, I've gone to emojis. So only this third wave feedback is inhibited by anesthesia, selectively preventing consciousness. This was shown for all different types of anesthesia by George Mashour's group. And so it's this third wave. What's so special about this third wave that results in consciousness? It does suggest that the mechanism of anesthesia holds the key to understanding consciousness. Now, when these signals get to the cortex, there's another 3 waves. The inputs go to layer 4, and from 4 they go to 1, 2, and 3, and 6, and 1, 2, 3, and 6 converge on layer 5 pyramidal cells. I think both speakers mentioned the pyramidal cells, which seem to be the best bet for where consciousness is going to occur. Not that it might not occur elsewhere, but because the apical dendrites from pyramidal cells give rise to EEG, which gives us the best neural correlate of consciousness, for example, in gamma synchrony, the pyramidal cells are the best bet for where consciousness might be occurring. So there's the Bing, and there's the emojis. So the other 4 are basically cognitive architectures, Anil said that, and don't address the hard problem, binding or memory. Memory is usually attributed to synaptic proteins, but they are very transient and last hours to days, and yet memories can last lifetime. Free will is impossible. Consciousness comes too late, at least for real-time conscious actions. Thus, real-time consciousness is an illusion. We'll hear about this later in the week with Aaron Sugar's talk in the panel to follow. And they view neuronal states as inanimate bits, consciousness and cognition emerging from complex network activities. And Anil brought up the relationship between consciousness and life, which I think is a key one. But the other models tend to treat neurons as simple on-off states and are somewhat of an insult to neurons when you think about what single-cell organisms can do. For example, the paramecium can learn, avoid predators, find food, find a mate, and have sex. No synapses, one cell. And there on the far right in the blue is a blue movie, you might say, or an X-rated picture of 2 paramecium having sex. And we don't know if they're conscious by Bing, but okay, maybe I've gone too far. So how does the paramecium move around and perform its cognitive functions? Well, it turns out that there are extensions on the surface called cilia, which are shown here in cross-section. They're made of 9 pairs of microtubules, part of the cytoskeleton with an internal core, which stick out from the surface and can bend back and forth. And here's a close-up view of a centriole, which has a very similar structure. And now, how do these things bend in a coordinated fashion? Well, back in the 70s, it was, okay, so here's the cilia, and here's one microtubule, and it has these motor proteins, dynein, which use ATP to contract. And this causes the two adjacent microtubules to bend in a coordinated fashion. And a biologist named Jelle Attema in 1974 suggested propagated conformational changes along the microtubule triggered each dynein to contract, so it worked in a coordinated, purposeful function, and also could transduce sensory information. So how is this relevant to the brain? Well, it is actually. If you look inside a dendrite of a neuron, so here's a cortical neuron, here's the axon, here's the dendrite, and here's one microtubule inside. And if there's a synapse over here that needs a dopamine decarboxylase or some enzyme for its maintenance, for synaptic plasticity, synthesized in the cell body back here, it must be transported along the microtubules by these motor proteins, dynein and kinesin. And tau protein, which is a microtubule-associated protein, turns out to actually signal the motor proteins where to get off. It's like a FedEx code. Because these things have to jump from microtubule to microtubule since they're interrupted. And when you come to a branch point, you have to know where to go right or left. So it appears that the tau is like the FedEx code for delivery of synaptic cargo, and therefore important for memory and cognition. Now, if the microtubules disassemble and the tau falls off, that's Alzheimer's disease. And it's unclear whether the tau falls off first, and then the microtubules disassemble, or the microtubules destabilize, and then the tau falls off. And how this relates to the amyloid plaques is unclear. We'll hear about this from Rudy Tanzi tomorrow. This is also the same lesion in postoperative cognitive dysfunction. So when you lose the microtubules and the tau placement, you lose cognition and eventually consciousness. So if we look at the microtubules inside a giant pyramidal neuron, our best bet for where consciousness is, we can see they're interrupted and they're mixed polarity. Now, they're part of the cytoskeleton, so if you expect them to provide structural support like our bones, why would you break them up into little pieces? It doesn't make any sense. Our femurs, our long bones are continuous. They're also a mixed polarity and adjacent to each other. And we showed years ago that this is optimal for information processing and actually gives rise to resonance and if they're resonating, beat frequencies at slower frequencies. I'll come back to that point. So one point is that microtubules seem to be in the right place in the right time to perform cognitive functions inside neurons. And it was proposed by the famed biologist Charles Sherrington in 1953 that they might be the nervous system within the neuron, within the cell. And I started publishing on information processing in microtubules in the 80s, and eventually on quantum information processing with Roger Penrose. And so I've been kind of obsessed with microtubules for over 40 years. Now, they appear to play a role in memory, too. As I mentioned, the synaptic proteins here are transient, last hours to days. And one thing that, and I'm suggesting they have something to do with memory might be encoded at a deeper level, like in the microtubules. One thing that happens in long-term potentiation is that calcium comes in and activates this enzyme, CAMK2, which can phosphorylate 6 or 12 phosphorylation sites. So it's a very beautiful molecule. It's a hexagonal molecule. When calcium comes in, it sprouts these kinase domains and the phosphorylation sites, and then phosphorylates something inside the neuron which can encode information. So we got the idea that might be microtubules, and in this paper a few years ago with Travis Craddock and Jack Tuszynski, we noted and showed that the size and scale and geometry of the CAMK2 fits perfectly with the microtubule lattice. And this is our suggestion that the CAMK2 comes down, lands, and leaves an imprint, a memory trace, and this could be used to encode information in the microtubules for where tau might bind, and is I think, the best suggestion for memory that there is. It can't be in synapses because they're too transient. Microtubules and dendrites are stable. They don't have to be depolymerized for mitosis since neurons don't divide. They're capped so they don't disassemble. They're pretty much stable and permanent, and good locations for microtubules, for memory, excuse me. And it's also been noted that the patterns that we see in automata models in microtubules are similar to the structure of language as suggested by Chomsky. Noam Chomsky suggested that structure and grammar has language and grammar have a structure that must be encoded and instantiated somewhere in the brain. But nobody's come up with how that could happen in neural networks. And Massimo Piatelli-Palmerini, who's a linguistics professor here, and his colleague David Medeiros have worked on these X-bar structures, which are one of the ways that sentences and grammar and language can be shown in graph form. And we noticed the similarity to the patterns in microtubules, so I suggested to Massimo that maybe Chomsky might like this idea. And he said, you should tell him, which I did a few weeks ago when he was here. And he rather liked the idea, and we intend to pursue that over the summer, and we'll see where we get with that. So microtubules can, in principle, store memory, be the basis for logic and grammar, and could they be the site of consciousness? Well, I showed you this picture before, and the microtubules do add a much greater capacity for information processing to the brain. So if we look at the brain computational capacity at the level of neurons, as espoused by AI people in the singularity, with 10 to the 11th neurons, 10 to the 3 synapses per neuron, about 100 hertz, gives you about 10 to the 16th operations per second per brain. And people like Ray Kurzweil have been saying for years, well, when we get to 10 to the 16th operations per second, we'll have brain equivalence, consciousness. You can put your consciousness into a computer. Don't worry about it. Give us lots of money, and we'll fix it for you. However, if you consider microtubules as the fine grain of information, you get you get 10 to the 11th neurons, 10 to the 9th tubulins, microtubule subunits per neuron, at 10 megahertz. I'll show you the evidence for that in a second, which gives you 10 to the 16th operations per second per neuron, the same as what they're saying for the whole brain, or 10 to the 16th operations per second per brain. So that pushes their goalpost way downstream, so they don't really like that very much, and I'm rather unpopular among AI people. But as someone once said to me, okay, wise guy, let's say you're right, how would that explain consciousness? You know, joy, love, feelings, what later came to be known as the hard problem, thanks to Dave Chalmers. But this was before I knew him, at least, and I realized I didn't have an answer for that. But fortunately, I read a book by Roger Penrose, which was suggested by the same person, who unfortunately I forget who it was, that quantum computing of some sort might be necessary. And I read this book, The Emperor's New Mind. It was an attack against AI. It argued that consciousness was not a computation. There was some non-computable function, and that it required some sort of collapse of the wave function. Now, excuse me, I don't have time to go into this. We'll hear more about quantum effects later in the week. But the basic problem is in quantum mechanics is that things can be in multiple states or locations at the same time. For example, a cat can be both dead and alive if his condition is amplified or coupled to a quantum event inside a box. And it appeared from early work in quantum mechanics that the superposition remained, and the cat would be both dead and alive until the box was open and someone took a look. That consciousness caused collapse of the wave function. The cat was both dead and alive until someone observed it. And this was put forth by Wigner, von Neumann, Stapp, and recently Chalmers and McQueen. And we'll probably hear more about that later. Now, Roger Penrose had turned this around 180 degrees. He said, no, collapse causes consciousness, or collapse is consciousness. And he began by treating superposition, taking it seriously, combining it with general relativity, and saying a particle is equivalent to mass curvature, to curvature in space-time geometry. And a particle in two places at once is a separation or a blister in space-time geometry. So the universe itself begins to separate the structure of the universe. Now, if this were to continue, this would be consistent with the multiple worlds hypothesis, that you'd have two or more universes. But Roger proposed that these superposition separations were unstable and would self-collapse or undergo quantum state reduction at an objective threshold, thus OR, objective reduction, at a time t equals to Planck's constant over the gravitational self-energy, basically the indeterminacy principle. So that here's the superposition progresses, but it reaches threshold at time t equals h over g, over e sub g, and bing happens. And this one continues, and that would be a happy face, hopefully. And this is because, now the selections are not random as in other forms of quantum mechanics, but influenced by Platonic values in the structure of space-time geometry. Yeah, you could have a sad face. This one was not chosen. And why we would tend to choose happy things or good things in general was because, Roger suggested, the choices were influenced by Platonic values embedded in the structure of space-time geometry. Or another way to put that is resonating with the structure of space-time geometry. Now, most of these would be random and meaningless, proto-conscious, kind of like notes and tones of an orchestra warming up. You know, that you come in and they go, every instrument is playing its own, they're not connected. And then all of a sudden, the orchestra begins to play and we hear Beethoven or a rock opera or whatever type of music you like. And so I think these random events are occurring everywhere in the universe as like decoherence or OR. But it takes the brain to put them together to make music, to take noise and sounds and make organized music. And this mechanism, although it's been criticized and ignored to some extent, is the only mechanism ever proposed for consciousness. The only specific physical mechanism ever proposed to produce qualia and conscious experience. There's a lot of stuff about emergence and this and that, but it's not connected to biology or to anything in the brain. Now, how would this be connected to the brain? We'll get to that in a second. But the basic idea is that the qualia of the redness of the rose is not so much due to a pattern of metabolic activity, as in the upper slide, but it's because the particular patterns in space-time geometry, not just three, but many, many more, of the redness of the rose entangle and are recreated in her brain in some way, specifically in microtubules, I'll argue. So how could that happen? How would the random OR events be orchestrated in the warm, wet, and noisy biological brain? We heard for many years that you guys are crazy, the biology's too warm. But then about 10 years ago, it was discovered that plants use quantum coherence. And in fact, in the basic process of photosynthesis, in which plants collect photons in the light harvesting complex, and the energy is converted to electronic excitations, or excitons, and passed through to the reaction center where they're converted to sugar to make food. And this transfer process has to be very, very efficient to make food. And it turns out that the electronic excitations pass through a series of chromophores simultaneously. The energy goes through all of them at the same time. And therefore, you get the fastest and the most efficient transference of energy. And people looking for solar energy panels are trying to emulate this. So there's some kind of quantum effect going on in these chromophores, which are basically pi resonance groups. So that brings us to the basis for organic chemistry and life, carbon-based life. So we know that hydrocarbons are chains of carbons and hydrogens, which may have a double bond. And chemists in the 1800s were puzzled by a compound, benzene, C6H6, which didn't fit the formulas here, and they didn't know its structure. But then Kekulé, a German chemist, had a dream that the alkanes, which he saw as snakes, one of them swallowed its tail and became a ring, and he woke and said, aha, benzene is a ring. And it turns out he's exactly right. Now, what this means is that there are 3 extra electrons in a benzene ring. And this is the basis for all organic chemistry and life. Now, in this benzene phenyl ring, you have the extra pi electrons form basically an electron cloud. So it's a quantum distributed electron cloud which delocalized to form these pi resonance quantum states. Now, if you put 2 of these pi resonance groups together, they tend to attract because the electron cloud in one repels the other, and they oscillate back and forth at the van der Waals radius. They oscillate back and forth in terahertz, 10 to the 12th hertz, which is pretty fast for biological scales. But as we'll see, that could be very relevant. And this could be, you could couple several of them, as we think happens in microtubules, which I'll come back to. So the dipoles are oscillating back and forth among pi resonance clouds in proteins such as microtubules. So pi resonance has something to do with consciousness. And for reasons I'll tell you more about in a second. But all psychoactive molecules, serotonin and dopamine, the pleasure neurotransmitters and mood neurotransmitters, have pi resonance groups, as do all psychedelic drugs, LSD, DMT, psilocybin. We'll hear more about those later in the week, are basically pi resonance clouds. So they have all this delocalized pi resonance that can be go into dipole oscillations and superposition states. We also know that these benzenes can propagate quantum spin between quantum dots, as shown in 2003 by Auschalam's group, and this is actually increased by temperature. So rather than temperature destroying quantum coherence, which everybody was saying, through benzene rings, it's actually the quantum effects are increased by temperature. Okay, so what does this have to do with life? So these pi resonance clouds, like benzene, are often occur with polar tails on them, like dopamine. Dopamine is one, and these are called amphipathic biomolecules. And oil and water don't mix, so the benzene rings kind of stick together, and this is how proteins form. The nonpolar amino acids stick together, the polar ends stick out into the aqueous environment, and you have a nonpolar hydrophobic pocket inside proteins. And this self-assembling and protein folding forming these nonpolar hydrophobic pockets inside proteins. And the same thing happens in lipids, where you have the to form membranes, we have the nonpolar in the middle. And in nucleic acids, like DNA, there's a pi stack. So DNA has this quantum core within it. So there is, we argue, a quantum underground pervading living systems. And Travis Craddock, Jack Jasinski, and I have a chapter coming out called The Quantum Underground, which is where I got that term. So one other thing about these pi resonance groups is that when you put them together, they only can be stable in really two different forms, in T and P. They're not really stable in the S forms. And so you have two alternate possibilities. So if you imagine a collapse, a Penrose objective reduction, to one particular configuration, that might give a good feeling, like down here, and if they collapse to the other, that might be a bad feeling. So obviously I'm anthropomorphizing here, but qualia and feelings have to come from somewhere. Emergence, I don't think makes the argument, and I think that these are very proto-conscious, primitive. We call this proto-pleasure, Roger and I did in a recent paper. So this actually impacts on the origin of life. If you imagine the primordial soup where life began, we could have had these micelles which reach a threshold for OR collapse, and there's a simple bing, or a conscious moment, connected to space-time geometry occurring in the primordial soup, suggesting that primitive pleasure sparked the origin of life. We don't really know what caused the origin of life. Darwin's evolution doesn't really cover origin of life, and how did it get from the origin of life to having genes anyway. Now, most of these would be primitive and random and proto-conscious, but some would be positive and feel good, and this could give feedback to arranging the pi stacks to optimize pleasure. That's the argument I make in this paper that will be coming out soon, The Quantum Origin of Life, How the Brain Evolved to Feel Good. And it challenges neo-Darwinism in terms of survival of the fittest, because why would an organism care about survival if it didn't have any feelings? And if you say, well, it's programmed to do so, then you say, well, who's programming it? And then you're going closer to intelligent design. This is kind of in between, where it's kind of instead of a stick driving it, it's a carrot, and self-organization occurs to optimize pleasure. And so I make the argument that life was, the origin of life was sparked by these proto-pleasure events, and this also promoted evolution to the point of we are here conscious and thinking about consciousness. Okay, is there any evidence for all this? Well, I argue that there is, and it has to do with general anesthesia, which I mentioned previously can provide the best understanding of consciousness, and general anesthetics selectively erase consciousness, sparing non-conscious brain activities. How do they act? Well, over 100 years ago, they knew that there were many gases that caused anesthesia in all animals, and when they did, the same concentration was needed to put a horse to sleep, a salamander to sleep, a fruit fly to sleep, or a human, the exact same concentration. And Meyer and Overton in Germany and England looked for a common factor of what determined, and all these gases were different structures. They were chemically diverse. What was the common factor? It turned out it was solubility in olive oil. So here's 1 over potency, and the more potent they are, the more soluble they are in olive oil. And olive oil is, here we see a ground-up body, you might say, with the solubility phases, with consciousness, the Meyer-Overton correlation occurring at the most nonpolar region, which correlates with benzene and methylbenzene and dimethylbenzene, which is basically olive oil. So these are also similar to what's happening in aromatic amino acids inside proteins. So in the 1980s, Franks and Lieb showed that anesthetics, first they thought they act in lipids, and it turns out they act in proteins in nonpolar hydrophobic pockets of pi resonance clouds in proteins. So there's a hydrophobic pocket of aromatic rings, and the anesthetics act in this quantum underground in proteins. Okay, which proteins? Well, we just had an excellent morning session on anesthetic mechanisms, and most people study anesthetic binding to membrane proteins in particular, for example, GABA receptors, and we know that the anesthetics do bind to GABA receptors as well as serotonin receptors, glycine, acetylcholine. These are all on the surface of the membrane. And propofol etomidate also to GABA, and ketamine to NMDA receptors. However, if you look at the actual experiments on anesthetic effects on, for example, a GABA receptor, which is inhibitory, you get a mixed bag. Sometimes they go up, sometimes they go down, some don't actually bind to anesthesia. So that hasn't worked out. And in fact, leaders in the field have suggested we need to try something else. So in fact, some people have, and a number of years ago, Eckenhoff's lab at Penn, without any bias towards microtubules or anything, looked at binding of radiolabeled halothane to 74 to mouse brain, and then did a gel, ground up the brain, and analyzed which proteins the anesthetic bound to, and they found 74 proteins, half in the membrane, half in cytoplasm, including tubulin, the component protein of microtubules. They did proteomics and genomics, which pointed to microtubules mediating the functional effect. And in a follow-up study, they used a fluorescent anthracene anesthetic, which is only anesthetic when you fluoresce it, and they gave it to tadpoles, and who fortunately conveniently have transparent heads, so you can illuminate their brains with ultraviolet light, fluorescing the anthracene and making them go literally belly up. They would become anesthetized. They then looked at their brains, which were donated to science, and found that the anesthetic was bound to microtubules. And they graciously said this was consistent with our theory, at least one version of it, which they cited. So if you look at tubulin, the microtubule protein, here it is. It has 86, that's a lot, 86 aromatic rings. And here in the red, we see binding of halothane in this quantum underground, you might say, adjacent to these aromatic rings. The anesthetics bind nearby by Meyer-Overton, and because the arrangements of aromatic rings might correlate with feelings, there are so many aromatic rings that we have a huge number of possible feelings in one tubulin. And you multiply that times the number of tubulins in a microtubule, and then the microtubules in a neuron, et cetera, et cetera. But then you're going to get quantum effects, and they're going to become unified and so forth. So the point is that there's a great potential at that level. Now, Anirban Bandyopadhyay, who will speak tomorrow, has done some amazing work with microtubules, including this study, where here, can you see that? I don't know if you can see my cursor. So in the upper left, you see two neurons, 1 and 2, with 5 nanoelectrodes. And Anirban used 2 pairs of electrodes and applied various frequencies across the first neuron, and then looked for effects in the second neuron, possibly ephaptic or possibly spiking, probably ephaptic actually, and then plotted the frequencies which caused an effect in the second neuron. And then he did the same thing at the level of a single microtubule and of tubulins, and got this amazing data where you see going from the slow end to the large end, resonances in hertz, kilohertz, megahertz, and then you go down to individual microtubules, kilohertz, megahertz, and gigahertz, and then going smaller and faster, megahertz, gigahertz, and terahertz. So in terahertz, we're back to the pi resonance oscillations. And so we think that what's happening is that we have pi resonance oscillations at the slow end that are cascading, at the fast end, at the small end, cascading upward in scale and slower to reach even EEG levels. And we predicted that EEG is a beat frequency of what's going on at faster frequencies. So in recent work by Travis Craddock and Jack Tuszynski and their students and myself, we modeled a system of dipole oscillation. So here's a benzene ring in neutral position. And here's where the electron cloud is to the left, here's to the right, and it's going to oscillate between the two in terahertz. And we calculated that the average frequency should be about 68 terahertz at ambient temperature. So you've got 2 benzenes, they're going to oscillate at 68 terahertz. Now what happens if you stick an anesthetic nearby, which is what we did in the simulation? And we put a halothane 4.5 angstroms away, which is the distance we found in our tubulin simulation, and expecting to see perhaps that the anesthetics slow down the oscillations, but in fact, they increased the oscillations. And they raised it from 68 to 81 in this particular study. And, but that actually works out because that exceeds the available energy. So what happens is the oscillations become faster, but there's not enough energy to drive it, so they eventually stop or dampen. And this would make sense from the standpoint of anesthesia, because anesthesiologists know that if the level of surgical stimulus changes, there's more energy to deliver to the brain, the patient tends to wake up. So as the stimulus increases, we have to increase the anesthetic concentration. And this would explain that by increasing the energy to meet the frequency that it's been shifted to by the anesthetics. Okay, well that was for halothane, and recently we looked at a whole bunch of anesthetics and came up with essentially a Meyer-Overton correlation for anesthetic effect on pi resonance terahertz vibrations. So we plotted on the left the oscillation frequency, and the frequency as you go down the y-axis goes higher, which would actually be more anesthetized because it would exceed the energy barrier, versus the oil-gas partition coefficient. So this is basically a Meyer-Overton correlation for anesthetic action on affecting oscillation frequencies of pi resonance oscillations. And our next step in this endeavor is to actually, Travis has already started modeling this for all the aromatics in a tubulin, all those 86 and the different angles and different ones. And the first pass, he said, it looks a lot like what we got for individual benzene pairs. And if that occurs, then we'll try it for GABA, and we would predict that it wouldn't work for GABA. So we'll see how that works out and report back at a future date. So to summarize that part of it, anesthetics appear to act to prevent consciousness by effectively dampening terahertz quantum dipole oscillations in brain microtubules. So you might say, well, who cares about terahertz? I'm in the hertz range or milliseconds, much slower. So what we think is happening is that down at the terahertz, we have these dipole oscillations, and anesthetics act here to block the dipole oscillations, or actually to increase them above energy threshold. And as we move to the, you don't see my pointer, as we move from right to left into tubulins and microtubules and eventually neurons, you get slower and slower and slower cascades. And I think consciousness can actually move along this spectrum, and consciousness is more like music than it is computation. And the brain and the microtubules orchestrate the individual OR events that would be occurring naturally everywhere around us, and like the sounds of the orchestra warming up, into something that's more like music, or is music, literally, conscious music, you might say. Now, one aspect of this, one implication, is that megahertz, for example, which you see under the microtubules, 10 to the, about 1 million hertz. If you stimulate the brain with megahertz, you might resonate microtubules. It might have some effect on consciousness. And megahertz in electromagnetics is radio waves, and we're not interested in doing that. But it's all in mechanical, it's ultrasound. So for the last several years with my colleagues in psychology, Jay Sanguinetti, John Allen, and several others who I'll mention in a second, we've been doing studies putting ultrasound into the brains of 300 up to now college students, showing increased mood, enhanced mood. So like 30 seconds gives about an hour of mood enhancement. And we've just started a study on Alzheimer's, because it was shown in Australia that ultrasound markedly improves Alzheimer's in rats, and in the lab stabilizes microtubules. So here is Jay giving ultrasound to a fake patient. That's really one of our OR nurses. And you can see on the screen her opposite skull, which tells us that the ultrasound is going through the brain, bouncing off the opposite skull, and coming back. And so it's definitely penetrating the brain, unlike transcranial electrical stimulation, which may not. And our early results are encouraging, which is all I can say now. But we are in clinical trials, and we're eager to get more patients in. And in fact, Chris Chan is doing a patient back at the hospital even as we speak. So let me conclude and say that OR quantum computations in microtubules inside brain neurons produce conscious moments, regulate axonal firings. And by the way, I heard about firings. I think consciousness is in dendritic potentials, not necessarily firings. And if there are firings, it has to be integration before that leads to the firings. It also regulates synaptic plasticity in EEG and can underlie global workspace, predictive coding, higher order thought, and/or IIT. Any or all of those could be correct. And in the end, I don't see a whole lot of difference between them. For example, you could have predictive coding in the microtubule because you have patterns going both ways. So Anil's predictive coding might be in the microtubules producing quantum effects, which would tie what he's talking about to consciousness. Primitive OR proto-conscious feelings resonate with space-time geometry. This is the Penrose mechanism. People think this is really strange and weird, but remember, it's the only mechanism for consciousness ever put forth. Such events in the primordial soup could have sparked the origin of life, evolving towards orchestration and consciousness and the brain. The brain evolved to feel good. That's the argument. Resonating microtubules, megahertz vibration with transcranial ultrasound may treat Alzheimer's disease and traumatic brain injury to make new synapses and enhance synaptic plasticity. So let me acknowledge my collaborators. With OR, it's Sir Roger Penrose. For the anesthetic action and pi resonance oscillations, Travis Craddock, Jack Tusinski, Aliana Aquino, Jordan Prito, and Kamash Sahu. For the TUS, Jay Sanguinetti, John Allen, Chris Chan, Lindsay Briner, Abby Behar Montefiore, and Victor Mendoza, and the microtubule research, Anirban Bandyapati. So with that, I'll close and thank you for your attention.
Yes. If I could ask our other speakers to come up. We're running behind because of a late start, but we need to have discussion. Our concurrent session starts at 5.
Can I recommend that somebody runs the mic rather than have people line up?
Okay. You can do it.
You do it.
Well, so, and we'll have our speakers come up, and I would ask anybody coming to the microphone, please be concise. We don't have a lot of time, and we want to get the most out of this discussion. Sir.
Stuart Hammeroff, thank you very much. Very clear explication of your ideas. I would like to object to one claim that you make, that you have the only proposed mechanism for qualia and subjectivity. I think there is a second argument that we have made, and that is that the sentience begins at the level of the excitable cell.
Of what?
begins at the level of the excitable cell and not at the quantum level that you're talking about. But what I would like you to address is a related question, and that is, if the collapse of the wave function as you have described it is the basis for sentient events, why are all of phenomena that are described as conscious so restricted to life forms to begin with and animal life explicitly, and not generally in a panpsychic sort of way everywhere, if it is in fact just the benzene-like aromatic molecules, which is the essence of it?
Okay, I understand the question. First of all, I'm not sure all excitable cells are conscious. But moving on to the more important question, the OR events that would be occurring everywhere in the environment, including in this table, in this chair, in the air, everywhere, would quickly entangle. Most of them are polar, so they quickly entangle with the polar environment and rapidly reach threshold and have a collapse which is random and meaningless because there's no cognition. There might be a little flicker of some subjective awareness which would be random, but it wouldn't be protected from the environment. When you get in a non-polar environment, such as in the Meyer-Overton quantum underground, as we described it, you are isolated from the polar environment, and the system can do quantum computations long enough, avoiding the environment, to do useful cognition. So when the collapse occurs, it has meaning attached to it.
Right. So you do need life forms.
We have to move on.
All right, this is for Stanley Dehane.
Could you speak closer into the microphone?
Okay. You mentioned that there were 7 ignition patterns, and or different brain states, I assume. And I just wanted to call attention to an abstract 133 where it's called modular consciousness, and there are 7 brain elements involved in that, making us a society of 7. Anyway, I thought that was curious. Any comment?
I don't think I ever mentioned 7 ignition states, but if I did, it's probably my pronunciation. No, I think that there are as many ignition patterns as there are contents of consciousness, basically. What defines the contents of consciousness, according to my view, is a fraction of cells being activated within this workspace and the rest being inhibited, probably a much larger fraction being inhibited. So the richness of the contents of consciousness relates to the number of differentiated states that are available, much more than 7, obviously.
Oh, that's too bad.
You said in one of your, Stuart, you said in one of your first slides that the mechanisms of consciousness can be best understood by using anesthetics. I saw a documentary about 20 years ago by the BBC about witch doctors in Haiti that use like powders made up of like pufferfish venom and I think like a poisonous marine toad and a tree frog, and they literally turn people into zombies. And I think there's like a 10%, 90% chance of the person dying, but a 10% chance of them going into a dormant state.
We hate when that happens. No, anesthesia is quite reversible. The gases float in. They don't form any chemical bonds. They don't form any ionic bonds. You turn off the vaporizer, and by mass action, they float out, and the patient wakes up. So it's completely different.
Okay. Well, in about 10% of cases or so, the person goes into like a dormant vegetative state where the heart only beats about once or twice a minute, and they get buried because all their relatives think they're dead. And then the witch doctor comes along, digs them up, and turns them into his zombie. And there was a guy that survived this. He was a zombie for like weeks and weeks and weeks, and then someone recognized him and kind of like pulled him away, took him back to his family, and he recovered. But he had no free will during this episode. He was literally acting under the commands of the witch doctor the whole time. Do you think some of these toxins that they use could possibly be used to help understand consciousness?
I'd have to know what they were, but there's the world's expert on zombies waiting to ask a question over there.
Sorry, this is for Anil on a different topic. In using the predictive coding framework as a theory of consciousness, you come up against worries about the perception-cognition distinction, which I'm wondering how you end up handling. So cases of, say, a perceptual illusion, stick looks bent in water, you know perfectly well it's straight. I mean, in some sense, your brain's best model of what's going on in reality is that it's a straight stick. Nonetheless, what's in our consciousness, or at least in our perceptual consciousness, is a bent stick, which is a level of representation that doesn't have any particular obvious motivation just from predictive coding alone. So it seems like you need some other doohickey in your theory to explain why there's this distinctive model of perceptual representation in consciousness. And it seems to me that's going to be where a whole lot of the theory of consciousness comes in. So I'm just wondering if you could say more about that.
I think what you say picks up on one of the standard weaknesses of the predictive coding in Bayesian brain theory, which is very difficult to falsify, because basically you can account for almost any situation by just changing the priors that you think are used to interpret this or that situation, this or that sensory data. In terms of this perception-cognition divide, I think that's one context where predictive processing tells quite a nice story, because it can deal with the ways in which what you might hold at the conceptual level can deeply influence your perception without relying on without making a clear, bright line distinction between the two. But it doesn't necessarily mean that there always has to be a single consistent interpretation of a situation at all levels of predictive modeling. So you could have some situations which are indeed these cognitively impenetrable situations that I think your example refers to, where you perceive some sort of visual illusion. You know that at another level that what's out there is different, but that doesn't reframe your perceptual representation. And that,
I think the issue is really what motivates the selection of one of these many levels of representation as the conscious level of representation.
Yeah, and
what you said was that was the brain's best model, but no, the brain's best model
yeah, and I think
is
that's
the one in cognition. So it's a second best model, and why that one?
Well, that's one of the empirical questions, which is the predictive coding story doesn't say a priori that it's only the highest levels which reach consciousness, because we don't experience the world as conceptual labels. It's the conceptual level is integrated with the phenomenal level. But nonetheless, there are aspects of the predictive coding story which don't reach the level of conscious awareness also. So predictive coding is going on all over the place. It's going on the spinal cord. And for me, that's an attractive part of it, that we can start to delineate, in a way, do the sort of neural correlate experiment, but instead of looking at this or that brain area or this or that oscillation, we look at this or that computation of the posterior, given some priors. And sometimes they might be part of this, the functional neural correlate of consciousness, and other times not. The question of whether there's an overall stable state that things settle into that represents the best guess across all levels, I think that's probably still the right way, but that doesn't mean it's consistent with evidence that is presented to the brain at all the different levels.
I think Stan also does.
Yeah, maybe just a comment on this, David. So there is a very interesting view of the Bayesian brain, which is that Bayesian computations, probabilistic computations, are unconscious, and what we become conscious of is a sample from this available distribution, not necessarily the best, but in fact just one sample. So if the distribution is very peaky, it's going to be close to the best, but if there are multiple peaks, then you're going to see sometimes one, sometimes the other. This can explain phenomena like rivalry, and I think in my book I explain there's quite a bit of evidence for this idea that we keep an unconscious distribution, we become conscious only of one sample.
One last follow-up to that. I mean, this also, it might be, we usually think about the variance of these distributions as just modulating what the peak will be. But that's also an open question. And there's lovely work on ensemble and gist perception and perceptual metaphors and things like this and peripheral vision that suggest, at least to me, that some of the things that constitute our conscious phenomenology are expectations about variance and not just how variance shapes the mean. So that's just emphasized that the different components computationally of Bayesian inference could all play a role in determining conscious content, and that's what makes it an exciting empirical program.
I think we're going to have time for two more questions because we have the concurrent session.
For Dr. Dehaene, on talking about sound signal, you mentioned that you plotted respond to signal versus listen passively. what exactly you mean with respond to signal?
Can you repeat the question?
You said that on working with sound in neuron, you plotted respond to signal, to sonor signal, versus listen passively. What do you mean with respond to signal?
To respond passively, you mean? I'm not sure I'm understanding the question, but I guess this is one of the current hot areas in consciousness research, is we want to see if the signatures of consciousness that we have generalized from conditions where subjects are actively responding to the stimulus.
What exactly, what you mean with active?
Oh, in our case, I mean, this is typical of psychology experiments. So you instruct subjects to click buttons or report verbally, either to do an objective task or to report subjectively what they've been experiencing.
So you mean an external manifestation?
Right, exactly. So a report. But an interesting area of consciousness research is to try to develop no-report paradigms, where there is no actual need to report, but we can just read out from the brain whether the subject is conscious or not. And it's a very interesting discussion, whether what I showed today depends very much on having to report, or whether some of it, as I believe, is still present, though in reduced form when the subject is just passively watching, for instance, a movie. By and large, I say the signatures continue to be there, but some of them are diminished, and this is an area for current research.
I saw some data about with classification and decoding. I was just wondering how well suited this method is for studying consciousness. I mean, I was wondering how important the information you decode is also important for the brain itself, for instance. Or, I mean, a bad example, you can also decode whether somebody saw a stimulus by decoding activity in the retina or something. So I was just wondering about what it tells us about maybe the mechanisms and stuff.
No, that's a wonderful question. I think decoding has a lot of power. It helps us also to make sense of, the larger and larger numbers of data that we are recording these days. But you're absolutely right. This is just correlation. We're finding a correlation between a code in the brain and a particular state that the subject is reporting. Now, this is not sufficient to demonstrate causation. That's why I was very happy to show that actually there are some people who are developing animal models that go all the way to optogenetic manipulation, where you can really tell something about causality. In humans, this is much more limited, although I suppose anesthetics or TDCS or TMS are ways to stimulate the brain and demonstrate causation, but of course very limited for very good ethical reasons. So you're absolutely right. Decoding is powerful, I think, and the fact that you have superimposed states that can be decoded in several orthogonal ways, it's an important statement about the fact that there are superimposed states in the brain, but it's not sufficient in itself to prove that there is a causal relation between the signals and consciousness, for instance.
We'll have two more questions.
I have a question to Dr. Hameroff about the evolution of consciousness and the studies with pyramecium. I've been myself using tetrahymine, another ciliate, to study chemotaxis. And I've been wondering whether the chemoattractants there are mostly aromatic amino acids and aromatic peptides. So this might be related to your interest in the benzene molecule and the aromatic signal there.
Yeah, I don't know much about tetrahymena, but I know that single-cell organisms have receptors on their surfaces for compounds with aromatic rings, and yet there's no synapses there. So they're responding to the environment. And I would think that, dopamine is a classic example, and it's the pleasure molecule, the reward molecule, and it's like a benzene on a stick. So, and serotonin has the indole ring, and all psychoactive drugs, I think with the exception of acetylcholine, have aromatic rings in them. So I think that's where consciousness is.
Yeah.
And our last question, sir.
Just a question for Stan. You showed us that in animals, in monkeys, there's conscious processing or neuronal processing during unconsciousness, but isn't this the same what we experience every night when we sleep and dream? Because our dreams have the same structure as our conscious processing, and even sometimes we solve problems we were thinking about when we were conscious in the state of unconsciousness. So I think that's just evident.
I agree that the study of sleep is one of the most exciting areas at the moment, and some states of sleep are dream states where there is an actual consciousness, so I will certainly not deny that. I think in most cases sleep is a loss of consciousness, and in fact if you analyze what's going on, it seems to be a morcellation of brain activity, both in space and in time, so there is no longer a continuity in the ability to create these metastable states anymore. What you find is a disconnection of local activity states that no longer form a global coherence. Now, the suggestion would be that when there is a reportable dream, this is different and there's much more coherent activity.
So with that, I'd like to thank our three speakers. I thank you all for your questions. And again, we'll be continuing this tomorrow afternoon where we hear about integrated information theory, higher order theory, and then a discussion of these 5 theories. Thank you all very much, and the concurrent start at 5 p.m.